Okay, the recording is going. So when we ended last week, we had started to learn about generalized linear models, what to do when your data do not fit in that neat box where we can assume normally distributed error, where we get that in situations like if our response variable is binary, a yes or no, like we had in the example data set, did the seed germinate or not? And there are also other cases where we can get not normally distributed data. Greg, can you please mute your mic? Thank you. There we go. Okay, so if you have skewed data, for example, with a lot of values at or near zero and a few really high values, that doesn't play well with a normal error distribution either. So that's the next example we're going to go through, and we're also going to extend our generalized linear. So far, in the first week we did linear mixed models where we had a mixture of fixed and random effects. Then we introduced to general linear models where we have, generalized linear models where we have non-normal error. We can combine the two into the wonderful and useful tool called GLMM, Generalized Linear Mixed Model. Generalized because it can accommodate all different kinds of error distribution besides normal, and mixed because we can include both fixed and random effects. So let's go ahead and work on an example of that. So I will share my screen because we're going to, what we're going to do is look at a couple of slides about the example data set, and then we're going to go and get back into SciNet and get everything back to where we were last time so that we can continue to work on this lesson 2 example. Is everyone good to go? If everyone's following me so far, can you please give me a thumbs up reaction? Okay, I see a few thumbs popping up, so it seems at least some people are paying attention, so that's good. Thank you, thank you. So I will share my screen. Okay, so here is our next example. We're going to do generalized linear mixed model to look at an experiment on everyone's favorite vegetable. Yes, that's right, turnips. And we're going to look at what factors influenced differences in yield of turnips. As I said, this is going to unite the tools we've learned so far, the generalized linear model for non-normal and the mixed model for fixed and random effects put together to make it a GLMM. And I have found in practice that when you get to these complex models that have, both random effects and non-normal errors, the Bayesian approach really outperforms frequentist classical statistical approaches, and you can get much better performance, more likely to converge and get the answer that you want with a Bayesian model. So this is where these things really start to be the most useful, I think. And the syntax in terms of the coding in BRMS, there's really nothing new because we've already learned how to include fixed and random effects in the model formula on the right-hand side, and we've already learned how to specify the response family, the error distribution. So we basically just do both of those at once, and that's how we write our GLMM code. The example data set that we're going to use is also from the AgriDat package. It was published in a study by McConway et al. And they wanted to know what factors influence yield in turnips. Now, yield is the weight of crop produced per unit area, that is going to be a non-negative value, right? You can't have negative yield. You could have 0, but it's always going to be either 0 or a positive number. That may not lend itself to a nice symmetrical bell curve, because oftentimes you have a lot of low numbers and then a few really high numbers. So you have this skewed distribution. This is by no means rare. In agricultural science, we see this kind of data set all the time. So I'm going to go ahead and leave our slides behind here for a minute and go ahead into SciNet, where we're going to log in, start a session, and get back to where we were last time. So I've already logged in to Series on Demand, so everybody should do that as well. And this is exactly what we have been doing every week. And you can either start a new RStudio server session this way, or you can go to My Interactive Sessions, and you should be able to, if you have your previous week's session, you can click on this relaunch button and it will start a new one with the exact same options as last time, so you don't have to go back and enter them in. So yeah, that would be the shortcut, but I'll do it the long way in case anybody is unable to do that. The long way would be to go to RStudio Server in the Interactive Apps drop-down menu. Make sure that it says SEA Stats Training. Make sure the Q is Series. Make sure this one doesn't matter. Make sure that R is 4.4.1, at least 4 hours, just to make sure we don't run out of time. 4 cores and 16 gigs, and we can use this -- reservation equals Bayes 2025 because we've reserved the node for today. And I'm going to click launch. And it should get out of the queue fairly quickly. And whenever, now we can connect to RStudio Server once the job is started. Okay, and hopefully if everything is configured properly for you within the Bayes project, once you connect back to RStudio Server, you should see that the Stan path was set so that the system recognizes where to find Stan. Let me actually make this a little bit bigger. There we go. If everyone is able to get to this point, either on SciNet or on their own computer, please give me another thumbs up just to make sure that people are following along. And if you need help, either raise your hand and unmute and ask, or Grant can also help you. Okay, I see that many folks are thumbing up. Yeah, if anybody is unable to, like, if you can't log into Series or something, please do let me know. Yes, Alan, do you have a question? Or are you confirming that it's good to go?
There we go. No, I was troubling unmuting. So I just wanted to confirm that when I reload, so I'm doing it on my
yes, sir,
own rather than Series. So I go to File, Recent Files, pick the one we were just working on last week.
Yep.
That brings it up,
Yep.
which looked fine. When it brings it up, does it reload the library programs that we need, or do we need to re-highlight those and load them?
It does not. So that's the next thing I'm going to do. Yep.
Okay, perfect. Thank you.
You can follow along just as we're doing. So yes, so I would say the next thing you should do is go ahead and scroll all the way back up to the top. And we don't have to run everything because the two examples are sort of separate. So I would just go all the way back up to the top. Your worksheet should still be open from last time. If it's not, go to Files, and go to the worksheets folder in here, and we're still on the lesson 2 worksheet. Mine is still open from last time. Yours should be as well, but if you had closed it, that's where to find it. Now scroll all the way back up to the top and go ahead and highlight all the code up to the options line at line 31. Everything up to line 31. And then I'm going to hit Control Enter to execute that code, which will load all the packages that we need and set the model fitting options. Okay, so we're good there, right? If anyone is unable to do that or gets a weird message, please let me know. And now we can skip past all the stuff we did last week. Let's see, we should be, I mean, your line numbers might be slightly different if you've entered in comments or anything, but mine is at line 148, the gamma generalized linear mixed model turnip yield data set. So that's where we're going to go. And the first thing that we're going to do is put our cursor on line 151 and hit Control Enter. Data is the function that loads the example data set from the package that we're going to be working with. In real life, we usually read data in from an external file, but here we're just using an example data set that comes preloaded with a package that we have installed. Okay. Now I'm just going to go ahead and type in my console head parentheses McConway dot turnip. That's the name of the data set, just so we can see what's in there. What are the different columns that we have for this example data set? We have a gen column for the genotype. They're comparing the yield of 2 genotypes. And this is a 3-way factorial experiment because we have 2 different genotypes. Apparently, they were looking at whether the time of year or the date that you would plant or sow the turnips, does that affect the yield? We have different dates there. I think there's 2 levels of date. And we have density. So what is the planting density? I believe we have 4 levels of planting density, 1, 2, 4, and 8. And this is a randomized complete block design. All combinations of genotype, date, and density are 3 treatments. All combinations are replicated 4 times in 4 replicate blocks. They're called B1 through B4. It's a pretty standard agricultural experiment, and the response variable is continuous variable yield. And we can, if you want, you can type $McConway.turnip in your console because all the data sets in this package have documentation that you can look at. If you hit enter, it will pop up in the help panel. Yep. So this is what I just went through. So you have that documentation and the citation for where it was published and that kind of thing. So first, let's go ahead and look at the distribution of yield data, ignoring treatments or anything for right now. Let's just make a histogram of yield just to see the shape of the distribution and what kind of numbers we're dealing with. So we'll use this line 152 in ggplot. The variable on the x-axis we're plotting is yield. That's the only variable. Histogram is a univariate plot. So just yield is the only variable we need to enter. And geom underscore histogram is telling ggplot to make a histogram of the frequency of the different yield values. So then I'll hit Control Enter to run that line. So you can see that yield is obviously very skewed. We have, they're all positive, right? There's no negative yields. Some are very near zero, if not exactly zero. They're very close to zero, and a high peak around 2 or 3, and we go up to 25. But yeah, it's not symmetrical, right? It's like a very skewed distribution. Skewed meaning asymmetrical. So we're already thinking, okay, this is not going to be amenable to a normally distributed error. Because if it's normal, the error is equal on both sides, which would kind of end up going towards the negative. We'd predict negative yields. That doesn't make sense. The next thing we're going to need to do now, density. You can see the levels of density are actually integers, 1, 2, 4, and 8 kilograms of turnip sets per hectare were planted, but we are just going to, for the purpose of this analysis, consider density to be a categorical variable. We're going to compare the 1, 2, 4, and 8 as discrete levels, even though, yes, you could try to fit a slope through that as a continuous variable, but that's sort of beyond what we really need to do here. We're just going to consider them as 4 discrete levels. And so, but right now they're integers in the data set. We need to convert that variable to a factor so that we can have it be discrete levels in our model. And so on line 154, I'm going to say factor. Mutate means change a column's value. And so within the McConway-Turnip data set, we're going to change density to factor of density to turn it into a factor. Then I'll hit Control Enter to run that line. Everyone good so far? Next thing we're going to do is we're actually going to plot the data, not just the distribution, but we're actually going to plot what it looks like for different treatments. Now, I'll have to go back to my notes here. Yes, so what we're going to do is we want to plot the density levels on the x-axis, the 4, 1, 2, 4, and 8, 4 levels of that. And we're going to plot yield is our response variable on the y-axis. And we're going to separate within each density. We're going to make a separate box plot to show the distribution for each planting date and use color to indicate planting date. And then we're going to, because there's that takes care of density and date, but we also have genotype. So then we're going to separate the whole thing into 2 panels, one for each genotype. So this is like a way to plot all of the 16 treatment combinations in one plot and show the distribution of the yield in each one with a box plot with a nice colorblind friendly color scheme and a nice set of labels to actually make the plot interpretable. So density, we're going to put on the x-axis. Yield is our response variable on the y-axis. The interaction of density and date, within each density, we're going to split it up by date. And we're going to color the box plot by date as well. And then the facet means we're going to split the whole thing into panels by genotype. Hit escape to get rid of the autocomplete, and then I will Control Enter to take a look at this plot. So what do we observe? Well, we can see, first of all, it looks like there's sort of an increasing trend of increasing yield with increasing planting density. That makes sense. We also see that that trend is not identical for all the genotypes and all the planting dates. So for instance, this genotype Barkant has extremely high yield if it was planted in later, but only at high densities. And Marco, we sort of see a similar trend, but not as extreme. So there's sort of like a 3-way interaction where you have the highest yield is only observed in certain genotypes under a certain density at a certain planting date. So you need all three, the interaction of all three variables to explain the variation in yield. So we have probably a significant three-way interaction in this study. But what else do we observe? Well, you can also see the variance within each treatment combination is not the same. Like for ones where yield is low, that box plot, the box shows the middle 50% of the data is super narrow, versus the ones where the yield is high, it's very wide and has long whiskers as well, showing where the data within 1.5 times of this range are found. So there's a relationship between the mean and the variance. Now, if we know our basic linear model theory, we know that a regular linear model with a normal distribution assumes there is no relationship between the mean and the variance. The variance should be the same in all the groups, which we don't obviously see here. So that's another red flag that the normal distributed error is not going to be a good fit to the data. If you know the term homoscedasticity, that's what that's referring to. Constant variance across groups. That's Bayesian versus classical statistics has no bearing on that. That is something that is assumed when you're fitting a linear model, regardless of whether it's Bayesian or not. It's, we're subject to that requirement. So that's like, oh, I'm looking at this, I'm seeing there's a relationship between the mean and the variance. Probably a normal model is not going to be good. But let's go ahead and fit one anyway to demonstrate why it might be a poor choice in this case. So what we're going to do is, here's our BRM, our model syntax. It's going to be yield is our response variable, right? So that is on the left-hand side of the formula, followed by a tilde. Then we have our fixed effects plus our random effects. So we're just doing a linear mixed model. We're not doing a generalized linear mixed model yet, because we're still assuming normal error. Our predictors are genotype, GEN, date, and density, and we want all the interactions between them. So we're going to do genotype asterisk date asterisk density. That'll give us the main effects plus all their interactions. And we're going to do a random intercept for each block, in case there is some systematic difference in yield between blocks, because the blocks represent different environments. The random intercept within these parentheses, we put a 1 type block. So a random intercept, 1 for every block. And prior, remember class equals B means we're going to put a prior on the fixed effects. And I'm going to put a sensible prior on this. Now, if you look at the graph, we have yield goes up to about 25. So to me, a normally distributed prior with a mean of 0, because we're not going to say which direction the effect is. It's going to be, we think there's just as much chance of negative as positive. And standard deviation of 5, which is pretty generous and encompasses pretty large effects in this context. So we can, and remember, that's only the standard deviation. We also are saying there's a non-zero probability of effects larger than 5 as well. But that's just keeping our prior within kind of the order of magnitude of the data. And here's our data set, and I've just put defaults for how many chains and how many iterations. And then I'm going to hit Control-Enter to run that. And because I have it saved as a pre-fitted object, it just runs instantly. If you're following along, please give me another thumbs up. I'm going to keep annoying you with the thumbs up because I just took a class earlier this week on how to teach live coding demos and online workshops, and they said to make good use of those reactions. So I'm going to force you to keep doing the reactions. So sorry in advance for that. Kind of make sure you're all alive and stuff. So let's take a look at the posterior predictive check plot, the PP underscore check, which we should kind of do as a matter of, we should just do that as a matter of routine, every model we fit, because it shows us, does the shape of the fitted values match the shape of the data? Let's do that in this case. It's going to show us the data as a black line and the posterior predictions, a set of random draws from the posterior as blue lines. Now, we can see it's not ideal, right? Because the data are skewed, just like we saw when we made the histogram. Our posterior predictions are pretty much symmetrical. Not to mention, they go well into the negatives. Now, this, just the way the smoothing is, it looks like the data go below zero. They don't really, but the posterior predictions go well below zero. We're predicting negative yield for some treatment combinations, which doesn't make sense. So it's not a good fit to the data. Let me see, back to the notes here. We've already done that part. Yes, I'm not sure that this came up in this case, so I'll just skip that for now. Yes, now we're going to let's just say density is the treatment variable that we care about right now, and we'll just kind of ignore genotype and planting date at the moment, and we're just going to get the average, the model's predicted average yield averaged over all the blocks, genotypes, and planting dates. Just look at those 4 density levels averaged over everything else, not worrying right now about the interaction, and see what we get in terms of predictions. So we're going to do EM means by density of this model. Okay, and I'm going to hit Control Enter to run that. And you can see that while the point estimates are all positive, we do get a negative bound, lower bound on one of the credible intervals for the lower yielding treatment. So again, that doesn't make sense, because it means we're 95% sure that given the data we've observed, that the true value of yield for that treatment is somewhere between negative 0.6 and 4.6. But that doesn't, I mean, that's not ideal. We'd rather have all those values be plausible values. So what do we do about this? Well, the old school way of dealing with this problem is, right, if the only tool we have is a linear model assuming a normal distributed error, and we have some data, we have to make our data fit the tool that we have. We have to make our data have a normal distribution error. Well, and have roughly equal variance in all the groups. How can we do that? Transform the data. And what kind of transformation do we do if we have a skewed distribution? Anyone? It's written, the answer is on the screen already, so all I have to do is read it off of there. What transformation might you do on the data to have it have a more normally distributed error?
Logarithmic.
I see I don't have the chat up. People are saying log. Excellent. That's right. Thank you. So that's just what we're going to do here. We're going to, you can just do the transformation right inside of the model. So all we have to do is type log parentheses yield as the response variable. By default, this is the natural logarithm, but it doesn't matter what base you use. And let's see, I cannot remember what prior I put on there, so I'll go into my worksheets already filled in. and take a look at that. Oh, whoops, we've already passed that part. Yes, so we're going to use a prior with a mean of 0. Again, we're not putting our finger on the scales in terms of positive versus negative, but a standard deviation of 2 we can actually get away with in our log linear model, because a 2 log change is a very large effect. So that's we can reasonably be sure that we don't have a high probability of observing effects of much larger than that. So we're just going to make that prior a bit narrower, because now we have different units. And that's all we have to do. So let's run that. Other than that, yes, so I have a question about making sure everything is greater than 0. So in this example data set, we don't have any zeros. We have only positive values that are greater than 0. There was no, just the histogram looks like some are zeros, but they're just very small non-zero numbers. So yes, if you have zeros in the data set, the old school way of dealing with it is to add a small positive constant to every number before you do the transformation. But there are actually better ways of dealing with it than that, which we will do next week. But right now we don't have to worry about it because there's no zeros. It's all positive values, right? Because log of 0 is undefined and log of a negative is undefined. You had to have a positive value to take the logarithm. Thanks for the question. We'll get to that next week. Yes, the seed. This is a random number seed, which, because NCMC, if you remember what we're using to sample the posterior distribution, is an algorithm that uses randomness to estimate an integral. And we, for demonstration purposes, want to get the same result every time we run that. So we're just initializing the random number generator at a specific value using the random seed. You could put any number in here. I just put in these, it's basically just a number I chose off the top of my head. And yeah, so if you are doing a Bayesian analysis for a publication, you want to include a random seed in the code, so that, because you want to be able to reproduce those results identically if you had to rerun the code. So anything that you're going to publish should include that, so that other people can also, if they ever want to check your results, they can reproduce it exactly as you did. Even though, right, if we do enough samples, the sampling error will eventually decrease to close to 0, and you'll get basically the same result regardless of what random seed you put. But it's better to, so that we can confirm everything works just as it should, we should use a random seed, if that makes sense. Thanks for the question. So I'm going to run this code by hitting Control Enter. And yes, so Adam asks a great question. Even if you put the same random seed, if you have a different version of Stan or a different operating system or a different version of R, you may not get the exact same results. It's sort of contingent on everything being the same. So that's another reason why, in terms of reproducibility, I always include as much information as possible about what versions of all packages and software were used, so that to minimize those issues coming up. So let's run the posterior predictive check on this log linear model. That looks a lot better, right? We've got, now this is on a log scale now, so some of the numbers are negative, which is fine. We actually have a very good good match between the data and what the model is predicting. That looks pretty good. So back to the slides. So we might say, okay, we're good with the log transformation. It's fixed the issues with the skew. The variance has evened out, but actually it's not really the ideal way to do it, right? We are made our data fit our model. When you do that, you have to transform and then back transform. And actually, a small bias is introduced when you do that. And it plus these transformations are sort of limited in what you can do with them. So I think it's a better practice to keep the data as they are on their original scale and pick a model that fits the data instead of trying to make the data fit the model. Let's pick a response distribution that actually looks like the distribution of error that we get. And a distribution you can use is the gamma distribution in this case. If I have positive and right skewed data, right, like it's got a lot of low numbers and a few high numbers and they're all positive, the gamma distribution fits that really well. Here's a I made a plot of a few gamma distributions with different parameters. Now, right, like a normal distribution, no matter what mean you put and no matter what variance you put, it's always a perfectly symmetrical bell curve. It might be skinny, it might be fat, but it's always a symmetrical. Gamma can be kind of symmetrical-ish, but it can also, depending on what parameters you use, become very skewed as well. You can see we have some very skewed gammas here. So that works well. Also, its variance is proportional to its mean, right? This one, red one has a low mean and a low variance. The yellow and green ones have a higher mean, they got higher variance. So actually it's perfect because not only can it be skewed, it can also have account for that relationship between the mean and the variance that we saw when we plotted the box plots, where the lower ones had both a low mean and a low variance, then the other ones had a high mean and a high variance. So let's try gamma. And the way we do that, now we're going to do a GLMN. It's got those fixed and random effects. And now instead of transforming yield, we're going to fit this non-normal distribution to yield, gamma with a log link function. So it has a similar effect to the log transformation because we're using log as the link function, but all that stuff is done within the model. We're not transforming the data to fit the model. That's all within the model. And since it's still kind of a log scale model, we can use our same log scale prior with only 2 standard deviation. So yeah, not log yield anymore as the response variable, just yield. The log is done as the link function. Gamma is the response distribution and log is the link function. What we're using to sort of dialogue between the data and the model, but it's done within the model. So yield and gamma, it's actually a capital G. That's just the way it's written in statistical notation. And link equals quote log. You can also use inverse as a link function, but we're going to use log in this case. Since we sort of showed that that's how you deal with the log can even out the variance if you have a relationship between mean and variance. And we're going to say normal 0, 2 parentheses as our prior. Control enter. Again, I use the pre-fitted model object, so it runs without having to compile and resample the model. Now let's look at the predictive check plot for this model. You can see here that right, the last one showed the transformed data, but here we have the original data. Right, the data have not been transformed. It's still that same skewed data, but our predictions are skewed in exactly the same way, right? So this is nice because we didn't have to transform the data and we got predictions that match its shape. So this is chef's kiss. This is exactly what you want to see on a posterior predictive check plot. So we're happy with that. Let's look at what predictions it makes. We're just going to look at density still. So we're going to, in our EM means this time, we're going to say density, tilde density means we want predictions for each level of density averaged over everything else. And type equals response indicates that we want those predictions on the data scale, not on the link function scale. So we'll get them in the units that that yield is in, not in the log units. which is kind of like back transforming, but because the transformation is done within the model, it doesn't introduce the bias that would happen if we did the log transformation outside of the model. So it's a little bit better to do it this way. Control Enter to run that. Results may be misleading due to involvement in interactions. Thank you for that note. I'm aware of that, but this is only to illustrate one main effect. We're averaging over all the other effects, but that's a very helpful note. And then I'll hit Control Enter here to print the output to my console. You can see that we have our different response means. They're quite similar to what we had before. Those are the posterior median estimates for each density level, and here are the upper and lower bounds, though, right? We don't have any negative ones because the model was fit on the log scale. So all those credible intervals are reasonable, plausible values. You never have any negative predictions anymore. That's exactly what we want. Let's see. Yes. Oh, and actually, yes, on the slides, I have that we would actually want to look at the estimated marginal means for every combination of treatment. And this is how you would do that. Density plus date plus genotype. You don't have to put asterisks. It will give you every combination if you do it that way. And again, but we are, the estimated marginal means will set the random effects to 0. So this is kind of like, we're not including the random effect in our predictions, only the fixed effects. It's the expected value for just a random block, that not necessarily for a specific block that we did our study in, but for a random block, because we're just trying to account for the effective block, so that we can look at our average treatment differences. So yes, we can modify line 195 and say density plus date plus genotype. Hit Control Enter. Now we don't get the note about misleading and everything, but now we get, right, we get 16 combinations, 16 means, because we have 16 treatment combinations. And we can now do contrasts if we want to do hypothesis tests to specifically compare different levels of density or planting date or genotype. And because this model was on the log scale, the contrasts are going to be ratios. They're multiplicative differences. So like we wouldn't say yield is, 2 kilograms or whatever per hectare higher. We would say it's 1.5 times higher or 0.8 times as high. Those are on the ratio scale. And so if we wanted, let's say we wanted contrasts between each level of genotype for every possible, between each level of density for every combination of date and genotype, this is how we would do it. And I do rev pairwise for reverse pairwise, and that will put the higher density on the numerator, which I think makes more sense, so that the ratios are greater than 1, so like 5 times higher, not 0.2. That's more interpretable. So we'll get contrasts between each level of density by each combination of date and genotype. Rev for reverse pairwise. So we had to put in quotes date and genotype. Contrast. Error in find by rows. Yes, so make sure that you I kind of made a mistake and ran this first line without including all the variables in the original EM means. So you want to modify line 195 to have density plus date plus gen as the variables that we created the different combinations for predictions. Then once you do this, it will work. Hopefully that will fix the error. So now if we look at the contrast on line, by running line 199, you can see we have all it's a lot of output, but if you were interested in the results of this experiment, you could be like, okay, if we do an early planting date for the Barcant genotype, there's the ratio of yield between these two density treatments is pretty close to 1, and there's not a very strong evidence for a difference. But like if we went and looked at the Marco genotype, and the biggest difference is like the difference between the lowest and the highest planting density, there was a sevenfold increase in yield between the two. And here's the 95% credible interval means we're 95% sure, given the data, that the true difference was somewhere between 3 times and 15 times. So that's a pretty big difference. And we can also do interaction contrasts, and I'll show you that might make more sense once you look at the output. If we do interaction, we want reverse pairwise contrast, and we also want reverse pairwise interactions. And I'll show you how to interpret that. So this is giving us, for each planting date separately, it's giving us the difference in the density effects between the two genotypes. So like, is the density effect stronger in Marco or is it stronger in Barcant? It looks like most of these ratios are pretty close to 1, indicating that the effect of density within each planting date is similar between the two genotypes. It doesn't matter that much which genotype you are, increasing density increases the yield. And, yes, there's very little evidence that one genotype responds more strongly to increased density than the other. And we could go in and use some of the functions we've learned in previous weeks to get Bayesian style p-values to go along with these with those comparisons. So if you wanted to test against a null hypothesis of no difference between genotypes, you could do that. And the last thing I'd like to show before we wrap this up and take a break is I've included in here mainly in the lesson materials for you to peruse, text description of how you would write this model up in a paper, like the methods, what you'd put in the results, and a figure as well. So here's what you'd write in the methods. Again, I won't go through it now, but just the basic idea is like you talk about how you fit the model and what the model structure was. You could include model equations if you wanted, but I just have it described verbally here, and then the different predictions you made and what kind of comparisons you did. And in the results, you could say things like, there was not strong evidence for a different effective planting density between the genotypes, right? You could refer to a table with that, and then you could, whenever you give effects, I usually like to give the credible interval in parentheses. Those are the predicted mean yields for a specific level of density and date and genotype. That's how I'd usually write it. And for the figure, I'm just gonna I'm not going to go through the code, but I'll copy it and paste it into the console and run it just so you can see what it looks like. But it's also in the lesson text as well. So this is like an example of what you could show in terms of a figure. Right now, we don't have the raw data on this figure. We have the modeled means. The black dot is the median posterior estimate, and I've shown several different widths of credible interval around that. And if you recall back to the data, this is also just a good illustration that our model is actually capturing the patterns in the data, because not only do we capture the different means, there also you can see the credible intervals are wider as you go to higher, so it's capturing that mean variance relationship as well. Yeah, let me go back a couple of plots so you can see that. Yeah, so the plot of the model predictions looks a heck of a lot like the plot of the raw data, which is great. It means our model is a good fit to the data. So what questions do folks have about this? Okay, hearing none, I will, that's the end of the lesson 2 material. Let's take a 10-minute break and come back at 3:03 Eastern or 2:03 Central, and we'll dive into some exciting content of lesson 3. I will be right back, but after I get back, I will answer any questions in the break. But I'll be right back. Thanks. Hey, I see, Joseph, you have a question. By the way, my leg fell asleep so bad, I had to just like stand up and shake it around for a while to wake it up again. But yes, yes, Tulsi, you are correct. In this model, we cannot extrapolate to a different value of density because we were treating density as 4 discrete levels, 1, 2, 4, and 8. We might as well have said A, B, C, and D. But if we were treating as a continuous covariate, yes, we could extrapolate, but I mean, do that at your own peril, right? Like you can't extrapolate indefinitely higher than the levels of density the values of density that you tested, right? Because who knows when eventually, right, the yield would probably flatten off after density went up high enough, but you're never going to know when that would happen if you didn't test some very high densities. Right, like if you look at the I'll share screen again. If you look at the yield, it kind of looks like it's already starting to flatten off when you go from 4 to 8, right? So it's like 10, maybe isn't going to give you that much more benefit. I would just guess just from looking at the data, but it's hard to really make a firm inference about that without, if you're extrapolating beyond what the levels that you had. Great question though, thanks. Yes, maybe I should try a yoga ball out. I don't know.
Hey, Clay, just a quick question on the results
Yes, shoot.
example you gave us. So I'm guessing, so you say there was not strong evidence for a different effect of planning density between the
Right.
genotypes. I'm guessing that's because it was like, what was it, 9.8 and 8. But like, what's the criteria to say they're like there's
Yeah,
weak
so
evidence for a
again,
difference, or like
like
what's the cutoff? You know what I'm saying? Like,
yes. Now, part of my like soapbox or agenda or whatever you want to call it with this Bayesian thing is to get people away from the thinking of cutoffs of what is there an effect or not, because that is completely arbitrary. And we can, right, like 0.05 is this conventional cutoff for significance on p-values, but why is it that way? It depends on, right, like it depends on the stakeholder or the practitioner's need of what they consider to be a sufficiently big enough difference to be actionable, So why should we sit here and set an arbitrary cutoff from our
yeah.
chair,
Well, let me
Yes.
rephrase. I feel like if I were to show this to one of my bosses, they'd be like, they
Yep.
would, the first question would be like, they're okay. You state there's not a strong evidence for a differing effect. How do you
Yes.
discern that based
Yes.
on these results you
Right.
showed me?
Well,
You know what I
again,
mean?
yes, because they have drunk the Kool-Aid as well, right? So let me go back. But you can. I'm saying that just to start out with, but you can generate hypothesis tests with something that resembles a p-value and apply thresholds to it if you want to. So let's go back. I'll share my screen again. Right. So basically, the way that I would base that inference on is here we have is there a difference of density effect between the genotypes? This ratio is how much stronger Marco responded to a change of density between 2 and 1 versus Barkant, right? The ratio is pretty close to 1. Our posterior distribution credible interval is got a lot of probability mass on either side of 1. It goes from 0.3 to 3. So there's like sort of our most probable estimate is right near 1, and there's substantial probability on either side of that, right? So that I wouldn't say that to a Bayesian that would constitute adequate
Okay.
reasoning to say that there's not a difference. Now, we could go and put a Bayesian style p-value on that. And I just didn't go through that step for this example, but you could. And so
Okay.
the way that we have been doing it in previous ones is you would sort of pipe the results of this into the function describe underscore posterior that we've been using, and you could do the PD test, the probability of direction. What fraction of the posterior distribution
Yeah.
is on the same sign as our effect? And you can say as p equals true to get it in the form of a p-value. So let's just run that and see what we get. It should be
Okay.
as dot p. That's the that is the, yes, that should be.
So
Oh
I'm
no, my bad.
totally on
Oh,
board.
wait
and
a minute. Sorry, I got that wrong. It is as underscore p, but the null hypothesis we're testing
Oh, okay.
against is that ratio is equal to 1. I put
Okay, I
the
see.
default is 0, so that was testing the wrong null hypothesis. So let's,
Uh,
once you run that, like here are our p-values that you could use and say, well, they're, not anywhere near anything that you would remotely want to use as a cutoff for significance. So we don't have evidence for difference. So
So you've
yeah, you
sold
could
me.
put
I'm
those in a
totally on board
manuscript.
with the Bayesian
Yep,
approach. I just for
yep,
me, I know it because I've run this before when I've given reports to my bosses. These are the type
right,
of questions they're going to come back to me with and be like, "No,
totally,
rework this and give me a
totally.
p-value." So that's the
Yep.
only reason I wanted to ask.
Yes. And so since we're still and these things can actually be very useful. Like maybe there is a cutoff that you want to use. So like this helps you do that. So I think yes, blindly applying cutoffs is not good, but thoughtfully applying them can be good. So, and also a lot of reviewers in applied science journals still require them. So, let's put
Yes,
in the
true.
p-values, but also put in those effect size credible intervals as well, so that people could make decisions based on them if they need them. That's what I would say.
Okay.
Yes, Alan.
Yeah, so along those same lines, I number one, I agree with you particularly about the 0.05. I've dealt with that for years because O5 was an economic number. It's not a
Yep.
biologically meaningful number for us. So I often write about whether or not a difference that I see is biologically
Yes.
meaningful. But
Excellent.
I'm struggling a little bit with how to take the numbers you have and like in the results, how you're writing the results and convert that to that same statement. I don't feel compelled to have to give a
Yep.
p-value,
Yep.
but I'm just not,
Yep.
it's not rattling in my head yet how I would convert that language into this shows that there's not a biologically meaningful
Yes.
difference between the two. And that's where I
Yes.
want to be able
So
to get
let's
to.
see. Yeah, I guess to me it's like a matter of effect size, it's like, well, for this genotype, whatever, like, this is kind of a three-way experiment, so it's sort of overcomplicating things, but like if it's like, okay, 8 density gives us this yield and 2 density gives us that yield, it's a threefold increase, credible interval is between 2 and 4, so we're like very confident that there is a true difference. Is that threefold difference enough to justify the however much more it costs to plant 8 kilograms per hectare density versus 2, right? And like, but like maybe the difference between 2 and 4 is also like just a little bit less than a threefold difference, right? So it's like, oh, we can actually get just as much yield
Perfect.
benefit without having to go all the way up to 8. So like it's just a matter of the effect sizes, right?
Perfect, just right there. That was
Yeah.
perfect because if you talk about, okay, now I got it. Now that's making sense. So thank you. That's
But it's
perfect.
like, you could also say, okay, the p-value of the difference between 2 and 4 is whatever 0.0 something, and the p-value of the difference between 4 and 8 is not very big. So you could base it off
Right.
that too if you, or
Yeah.
I'm sorry, the p-value here would be high. So yeah, you could base it off that as well.
Sure. Yeah,
Yeah.
no, and I get that. But for me, I've wrestled with that for years. And like I said, I tend to
Yeah.
not, I mean, I report their p-values because journal editors will not almost publish unless you give them a p-value.
Yep.
But I don't
Yep.
talk about it in terms of whether it's, I hate the term significant
Yes.
or not significant, but rather, is it biologically meaningful? This difference that I'm seeing biologically meaningful
That's right.
given my system, and that's the way I discuss it. So I like what you just said as a way of doing that same thing, but using my Bayesian graph in this case.
Yes, very good.
To get there. So thank
Yeah.
you.
I think providing more numbers and like more summary statistics like, you know, effect sizes and then hypothesis testing statistics like p-values is good, and we should provide them, but they don't have to be like what we base all our decisions on. Okay, well, yeah, this is a great discussion. I really appreciate Joseph's making a comment. These models are explaining the system behavior within the bounds of data, not giving a predictive relationship. Right, yes, because I mean, this is a randomized controlled trial to compare specific management regimes for turnips, right? Like it isn't, that's like its goal, you know, it's not so ambitious to like go beyond that. So the inference we make, it can make predictions, but only within the context of the experiment that we've set up, right? Like that's really all it's good for, which is very useful. But yes, it doesn't make predictions beyond that. If someone says it does, they're kind of trying to swindle you, right? Yeah. Okay, so let's we're having a great discussion. It's more important than like covering all the material, but we can go ahead and keep plugging away at the material. So you can go ahead and close out your lesson 2 worksheet. And you could save the changes. Now, at this point, I think it is probably a good idea to just restart your R session. So go to session, restart R. And then go to the regular worksheets tab, or I'm sorry, the worksheets folder in the files tab, and you can open up lesson 3 worksheet. So we're going to be working on lesson 3 in just a minute, but I will go to the lesson 3 slides, which you can also access on the workshop page. I'm going to open these up, and remember that all the lessons also have text, full text that you could read if you want more in-depth explanations in text form. I'm going to open these slides, and then we'll start talking about lesson 3. And we have some review of what we've learned so far. Again, we're now at part 3 of our crash course here, and you already know how to follow along. And just to, you know, so that you can feel good about yourselves and what you've learned so far, we've learned how the prior, likelihood, and posterior interact to give us the answers we want. We've learned sort of the machinery, a little bit of how Bayesian models work, that we have to use Monte Carlo to sample the integrals to get the answer. We've learned how to fit models, make predictions, and test hypotheses like we were just talking about. And the specific models we've covered, we've gone from linear mixed models to generalized linear models to generalized linear mixed models. Very cool. Very useful tools you now have in your toolkit. Basically, the remainder of the next this week and the next 2 are just to kind of give you more and more specific tools within the realm of GLMMs to increase your toolkit and the number of different types of data you can analyze and situations that you can work with. So it's just like kind of adding little bells and whistles to GLMMs in different ways, but not esoteric things that you'll actually come across in real life. And very common in ag science, speaking of real life situations, is that you have, you know, we're out there in the real world, things vary in space and time, right? So a lot of our studies, whether it's experimental or observational, we have to collect data at different times or at different locations in space, and there's variability in the environment in the world that occurs over space and time. How do we deal with that in our statistical models? That's what we're going to talk about today. And there's kind of two ways to do that, which I'll go over, and we'll give an example. We'll do an example of each. So the basic insight that you have to have to understand how these space and time models work is the closer together any two things are in space or time, or the space-time continuum, if you want to be nerdy about it, the closer two things are, the more likely they are to be similar, the more closely related they are. That's talking about it in a spatial context. They call that the first law of geography. The closer together two things are, the more likely more common features they'll share, the more likely they are to be similar. And same with time. And we're going to explicitly model that pattern of similarity decreasing as you get further apart in space and time in our stat models. Like here's an example that is, something that people do in ARS. Like people might want to look at how different farm management practices affect the level of soil carbon sequestration on the farm. But not all farms are equal. But the closer together two farms are, the more likely their environments are to be similar. They'll have similar temperature, precipitation, soil type, maybe even. Now, even if, farmer A and farmer B are completely independently decide what management to impose on their, what tillage and all of that kind of stuff to do, they do that independently, but they are not statistically independent if they are close enough together that they are sharing environmental factors based on being close together. They are not fully statistically independent because they have shared environmental influences from being so close together. And this is an issue, even in the most perfectly and carefully designed experiment, that's still going to be an issue because your experimental units have different distances from each other in space. And obviously in observational data, it's an issue as well. So the first way we can deal with this problem is by modeling the space and time relationship between data points residuals. So far, we've assumed that once we've accounted for all the effects in the model, fixed, random effects, all of that, once we've accounted for that all our residuals are completely independent of each other. All the models we've done so far have assumed that. After you plug in like if I have a observation and I know it's fixed effect and I know it's random effects, any difference in its response that's not accounted for by that is random noise. It's completely independent, random variable, has nothing to do with any other data point in my model. And if you are a SAS nerd, you know that the term G side, because in a model that assumes all completely independent residuals, the random effect variance covariance matrix is called G, so that's where all the random effects are. We haven't done anything with the residuals. They're just completely independent. But that assumption is decent in many cases, but it can break down and be wrong if you have strong variation over space and time. Does anyone have any questions on this so far? Excellent. So how can we extend the GLMMs that we've learned so far to model this variation in space and time? We can actually add some parameters to our model to model this structure in the residuals, residual covariance. In other words, we can say, okay, the closer that two data points are in space or time, the more close their residuals will be to each other. The residuals are more correlated based on the distance apart in space and time that the data points are. If you know SAS, this is called an R-side random effect because it modifies the R matrix which is basically the matrix that tells us the pattern of how the residuals or the errors on each data point are correlated with each other. But the concept is more important than whatever letter you use to describe the matrix. I always get the G and R confused in my head myself. That's one way to do it. The other way, right, so we can either model everything and then have some residuals and model the residuals as being correlated, or we can actually include the space and time effect in our model itself. Instead of modeling it in the residuals, we can actually include the term in the model. And you may have heard this called a smoother, or it could be called a spline. And it basically models the response variable as being a function of some smooth function of space and time. And that will make, we sort of left the world of GLMM behind, and we're now in the world of GAMS, a generalized additive mixed model. We'll talk about that more. That's our second example. We'll talk about that. But so there are basically two approaches. Model the space and time relationship in the residuals, or put it in the model itself using splines, making it a GAM. We'll go through both. And our two examples are, number one, our first example will be about time, and we're going to use residual covariance in our GLMM to model autocorrelation in time. In our second example, it will be over space, and we're going to use a GAM. But you could use either approach for either space or time, or you could even combine them both in the same model if you really wanted to get crazy about things. You could have a GAM with residual covariance as well. And again, the more complicated your model gets, the more likely you are to need the Bayesian approach with priors to actually get the answer you want to get. I'll pause so you can let that mind-blowing information sink in. So let's delve into our first example, looking at residual covariance to deal with repeated measures on the same individuals over time. This is very common in ag science, right? Because if you have plots that you take a measurement from at different time points, days, years, whatever, or animals that you're measuring at different time points, the measurements from the same plot or same animal at different days or different years or whatever are not independent. They all come from the same individual, so they must be related to each other. We have to account for that in our stat model. Otherwise, we'll have sort of pseudo replication and we won't get a correct inference. Yes, in a minute, I will switch to our studio server and we'll load the packages. But this is basically the same as before, except there's one other package we haven't used before, which is APE. And that we're going to use later to calculate autocorrelation statistics. Other than that, the packages are all the same as we've had before. Our example data that we're going to use for the first one, this poor cow in this picture is sadly afflicted with tuberculosis and isn't doing so hot. So in this, another example data set from the AgriDat package that comes from a publication by Diggle et al. They wanted to know if supplementing the cattle's diet with iron would help them still maintain their body weight gain, even if they had tuberculosis. So we have 26 cows, 26 individuals, some of which were infected with tuberculosis and some were not. I'm not sure if they purposefully gave the cows tuberculosis or if they just naturally had it or not, but that's not necessarily relevant for our statistical model, although it would affect the conclusion you could draw. But they did randomly assign within the infected and non-infected cows, they randomly assigned some of them to get iron and some not. Very simple design, 2 by 2. But the repeated measures part comes in because they measured the cows, each individual cow got weighed at 23 different time points, roughly evenly spaced. We're going to assume evenly spaced for the purposes of this analysis. Everybody with me so far? Please give me a thumbs up if you are following along. Thank you. So when we get into our studio, we're going to load the data, example data with the data function, and then I think it makes the most sense for the control level to be the first intercept level when you're ordering the factor levels, although the default is alphabetical. So we're going to use the re-level function to using the ref argument to set the reference level or the intercept level for each factor. No iron and non-infected are the intercept levels. I'll just and then the next thing we're going to do before we actually fit models is we're going to plot a individual trend line for each cow's daily weight. We're going to plot each individual cow's line will be colored by what iron treatment it got, and we're going to use solid and dashed lines to represent the infection status of each individual. That's the line type scale. And we're going to plot a separate line for each animal. And we're also going to plot a median, a median for each combination, each of the 4 treatment combinations, the 2 levels of iron, yes and no, and the 2 levels of infect, yes and no. So I will go in, just clear this previous stuff, and I'll highlight and run all the loading libraries and setting plot and model fitting options. Control Enter to run everything from the beginning to line 29. Everyone should do that as well. Okay. And now we can hit Control Enter to load the example data from Agridat package into our environment. And then again, as I said, we want to set the reference level to the intercept to be the control group in both cases. So we're going to use the re-level function. Ref equals no iron. And we're going to re-level infect as well. Mutate meaning create or modify columns in our data frame. Hit Control Enter when the cursor is there somewhere to run that line. Okay, now we're going to make a plot to look at our data before we fit any models to it. Day is the name of the column with the day number. Weight is the name of the response variable. We're going to color each line by the iron treatment. We're going to use the line type or dotted versus solid lines for the infection status. And group by animal means animal is the name of the column with the ID for each individual cow. So we want a separate line for each animal. So we'll make a line, geom line, and then we'll do this line will make a thicker line for the summary of each treatment combination. The median for all the cows in that combination. Control Enter. So here we see, you can see all the cows are gaining weight over time, over a period of several years. But it looks like you can see the solid lines are the healthy and dotted is infected. And you can see that obviously the solid lines are higher, so the weight tends to be higher for the healthy individuals. And orange is the control group, and blue is the iron supplement. So if anything, it looks like iron supplementation is is a little bit lower weight or a slower rate of weight gain, just from looking at this graph. Certainly, it's not benefiting much just from looking at the graph, but let's fit a statistical model to test that notion that we have from looking at the data. Okay, now, one thing that I think is something that you don't necessarily always have to do with classical statistical model approach, but is very useful in Bayesian, is to scale the predictor and response variables. Now, you can see that the day variable goes from like 100 and something up to 800 because they started the study when the cattle were already several months old. But I think it can be useful to for model fitting to make it more consistent and set all of the individual all of the variables so that we're 0 is the starting day for each cow. So we're going to make a new column called day after start by taking the column day and subtracting min of day from it. So we're subtracting the minimum from every value of day so that the lowest will be 0. But even more importantly, you can see that weight is in kilograms. It ranges from 100 to 500 or so. But I think it can be helpful to scale it to Z transform or standardize the weight variable so that it has a mean of 0 and standard deviation of 1. That is particularly important to do in Bayesian models because if you know that your response variable is in standard deviation units, you can set consistent priors across all your models. Like if you have a mean of 0 and standard deviation of 5, that's like 5 standard deviation units. You don't have to sit there and think about, well, the ranges by 500 kilograms. So should I put a prior of 100 standard deviation or 50 or whatever? Like it is helpful to standardize. Not to mention, Monte Carlo algorithms work better if the units, if the numerical values are not too big or too small. If we keep them in, a small order of magnitude, we can get better model performance. And it doesn't affect our answer because we're just subtracting a constant and dividing by a constant. It doesn't affect the distribution of the data at all. We can transform back and forth without introducing any bias. So it's fine to do that. The R function to do that is scale. We're going to make a new column called weight underscore scaled, and we're going to scale the weight variable. I'm going to highlight those 2 lines and hit Control Enter. So now our data are good to go. I'm going to go back to the slides and give you a little theory on residual covariance to explain the models we're about to fit. Please interrupt me if you have questions because this can start to be a little confusing, maybe. All right, so we just scaled the variables. Now, let's discuss what model we're going to fit. We're going to use as our response this standardized weight variable we just created. We want to know, is there an effective time? We're going to use day as a continuous variable, days since day 0, and asterisk iron, asterisk infect. So we're going to look at whether the slope over time, the rate of weight gain, the slope over time differs between the interaction of iron supplementation and infection status. Right, time is continuous. We're going to look at that in continuous units, and our 2 treatment effects are categorical in all their interactions. But that's fine, except that each of the cows has her own unique trend, right? They're all individual, and the measurements from the same cow on different days are not independent. So if we just fit the model this way without residual covariance structure, we would be ignoring the non-independence of measurements from the same cow on different days. And we would get sort of overinflated number of degrees of freedom. We would get the wrong inference about the effect of these treatments on weight gain. We need to account for the measurements from the same cow on different days not being independent. How do we do it? Here's a bit of math. So far, as I mentioned, we have assumed that after you account for all fixed and random effects, that the residuals are independent from one another. We call that conditionally independent because it's conditional on the fixed and random effects. The residuals are independent. There's still a residual variance covariance matrix in the model, where it would be, we call that R, the R matrix. But if you assume conditionally independent residuals, it is some constant, the variance, times an identity matrix. So basically, the diagonal of the matrix is the variance at each time point, basically. This is an n by n matrix, where n is the number of time points in the model. And it's a constant times the identity matrix. So basically, this constant is on every element of the diagonal. That means all the residuals have the same variance. But everywhere else, that matrix is 0. That means there's no relationship between any two residuals. So this would be like, this 0 would be the relationship between the residual of time point 1 and the residual of time point 2. This would be time point 1 versus time point 3. They're all 0. There's no relationship between any two residuals. They're independent. That's what we have sort of been doing so far. Now, the way we model correlation in time is taking the step of replacing some of those zeros with non-zero values. There are many ways to do that, but we're going to look at 3 of the sort of simplest ways that will be ones that work in most cases. The unstructured covariance matrix, compound symmetry, and first-order autoregressive. That's a lot of jargon. Let's explain what each of those things are. Unstructured covariance has the most parameters of any way to do this. Basically, we just say, okay, every single entry in this matrix besides the diagonal gets its own parameter that we have to estimate. So like this row of 1, 2 is the correlation of the residual of time point 1 and time point 2. And right, this is a symmetrical matrix, so the same value would be in 1 and 2, and 2 and 1 is the same. You've got, basically, if you have 4 time points, you'd have to do 6 parameters. 1 versus 2, 1 versus 3, 1 versus 4, 2 and 3, 2 and 4, and 3 and 4. Clearly, this is the most flexible and could give you the closest fit to the data because it has a separate parameter for every one. But it's got a lot of parameters. And look, 4 by 4, that's pretty easy, but you already have, we've got 20 something, I think 23 time points. So we would have to estimate hundreds of parameters, one for every combination of 2 time points. How could we simplify? Well, the simplest way would be to say, okay, every pair of time points is related by the same covariance parameter. 2 residuals, if they're from the same individual, they're correlated by the same amount, no matter how far apart in time they are. The correlation between time points 1 and 2 for an individual is exactly the same as the correlation between time points 1 and 4, for example. That's called compound symmetry. One other way to do it is first-order autoregressive. This means within each individual, the correlation between 2 time points in the same individual is higher the closer in time they are. You still only have to estimate 1 parameter, but every time you go apart by 1 step, you increase the power by 1. So like if your 2 time points are 1 unit apart, like time 1 and 2, they have this correlation, rho. If they're 2 units apart, like 1 and 3, they have rho squared, right? So like if rho is 1/2, then rho squared would be 1/4, and rho cubed would be 1/8. So as you get further apart in time, the lower the correlation. Eventually, it would be pretty close to 0. That's called AR1. Yes, so this is like a mathematical representation of the first law of geography, or one way of expressing it. The closer together in space or time, the more closely related. Exactly. Thank you for bringing it back to the concepts. We're going to do all three of these on our cow data just to see how they do. And remember, we have 23 time points. It was easy enough to look at the matrix with 4 time points. You have 6 parameters, but with 23 time points, there's actually 253 parameters. Often, in whether it's frequentist or Bayesian, if you have a model that's way too many parameters for the amount of data you have, it's going to have trouble. Whatever algorithm you use, even if you use priors, is going to have trouble fitting that model. And it just seems like a little excessive. So we can, if I was doing this in real life, I would probably not even try to fit the unstructured model because it's clearly way too many parameters for the data set we have. So really, it's a decision between is autoregressive or is compound symmetry a better structure. But I'm going to do all three just to show you. Here's how we're going to model the covariance structure in VRMS. You're going to use, there are different functions for the name of each covariance structure. So the unstructured one is called UNSTR, compound symmetry, COSY, C-O-S-Y, I always like that name. Autoregressive is AR. And then it has an argument time. That's the name of the variable that is which variable has the time in it. That would be day in this case. And GR is the name of the grouping variable, the subject. So that would be the individual cow. So we're saying, okay, correlation of repeated measures in time by day within each cow. Those residuals are correlated by unstructured covariance or compound symmetry or autoregressive. And this is how you would do it. They're all the same. The name of the function is different in each case. And first order autoregressive, we have to supply p equals 1. I didn't go over, so each time point is correlated with the one immediately adjacent to it. But you can increase that parameter to get more complex autoregressive models, but that probably isn't often something you would need to do for a simple repeated measures design. So we're going to use AR1 here. That was the model I showed you just now. And yes, let's go ahead and I won't go through the slide here. I'll just go ahead and start writing the models for you. So every model is going to be very similar in terms of the fixed effects. We're always going to have iron, asterisk, infect, asterisk, day after start. Those are the fixed effects. They're going to be the same for every model, right? We want our treatment effects and our time effect. For unstructured covariance, we're going to use UNSTR, unstructured. Time equals day after start. That's the name of the column in the data with our time. And GR for group equals animal. Correlated residuals within each animal. And we're going to use normal priors with standard deviation of 5 for our class equals B, our fixed effects. Again, because we've standardized in standard deviation units. Now here, this one is only something that you need for the unstructured covariance. We're actually going to put a prior on the entire covariance matrix to make sure that if I tried to do it without this prior and it failed to converge, because we need to put a little bit of a constraint on the covariance matrix parameters, again, because we're fitting 253 parameters. I needed to put a prior on it to give the model a little bit of information that we don't think the correlation is going to be too high. Otherwise, it didn't work. Actually, that's a red flag. If you have to do something like that, it probably means you're you've got too many parameters in the model anyway. But I'm sort of doing this as proof of concept. I also had to set all the initial values of every parameter to 0 because there were some issues with it in the model fitting process. So right, all these things, like if you have numerical issues with your model and Stan gets mad at you, it's probably because you're fitting a too complicated model for your data set. But I tried really hard to fit it anyway as a proof of concept. So I'm going to hit Control Enter and run that. If you're running that on your own computer, it will take a very long time to run. So I would recommend, if you're doing this on your own machine, either download the pre-fitted model object or skip this because it will take a very long time to run. Because it is not a very good model for the data set, so it takes a very long time to sample the posterior. Again, numerical issues caused by an inappropriate model.
Could have said that before I hit enter.
Yes, yes. You can always just like hit the stop sign to cancel it if you don't want, this is more for pedagogical purposes. So it's up to you if you want to like sit there and let it run. But on SciNet, we have the pre-fitted object. So you can see before I look at the output of the unstructured model, I'm going to go ahead and fit the other two because they're very similar to We're going to use the same iron * infect * day after start. The only difference is instead of unstir, we're going to use cozy for compound symmetry, the same time and same group variable. And oh yeah, I had to add extra warm-up iterations as well. That was another thing I had to do to get this unstructured model, which is way over parameterized, to converge. But I didn't need to do that for these other ones because they're more appropriate for the data that we have. I hardly ever would use unstructured covariance in real life. So with the same iron, by infect, by day after start are our fixed effects, day after start. And we're going to use the COSY, compound symmetry covariance. The time variable is the days after start, and the group GR equals animal. And I've called this Diggle compound sim for compound symmetry. Control Enter to run that. Same priors on the fixed effects. Lastly, the AR1 model. Again, it's almost exactly the same syntax, except instead of cozy, we're going to use AR. But we do have to add one more thing, which is P equals 1, because we want a first order autoregressive model. Other than that, the priors are the same and everything. Control Enter. Okay. Oh, yeah, and it gives me a warning that rows containing NAs are excluded from the model every time. I believe some of the cows died before the study was complete, so their time trend does not go all the way to the end, and they have a missing value. So those are just excluded. We're basically looking at the effect of iron and tuberculosis conditional on the cow having survived. So if we had a lot of attrition, we would actually have to think hard about how we would account for that in our model. But I think only maybe one animal didn't quite make it to the end. So we still have enough. The mortality was not sufficient to really throw things off for us. So we're just going to sort of ignore the fact that a couple of the cows didn't make it to the end, unfortunately. Let's go ahead and run the posterior predictive check plots to see if our, at least give us a quick check of whether the shape of the data is recreated by the model. PP_check. We'll do that on each model. Here's the unstructured model. That looks pretty good. There's sort of a bimodal distribution in the data, which I think are the healthy and sick animals, and it's capturing the shape quite well. How about the next one, compound symmetry? I'll hit Control Enter to run that line. It looks very similar as well. It's also the posterior is more or less the shape of the data. And here's the AR model. Again, we get that bimodal. You know, we don't see some weird skew in the data that's not captured by the model. So like all of them are doing pretty good. Now, in previous lessons, we have used leave one out cross-validation criterion to compare models, but that is not work very well for models with residual covariance structure, because leave one out cross-validation, if you remove one data point, fit the model, and predict that remaining data point, that only works well if the errors in each data point are independent. Because we're specifically saying they're not independent, the leave one out cross-validation will give you unrealistically good predictions, because there's actually information in the model that is sort of cheating and already knows a little bit about what that data point is before, because we're modeling them as not being independent. So we shouldn't use the leave one out cross-validation for anything that has a residual covariance structure in it. What we have to use is a cross-validation, k-fold cross-validation, where we're actually going to take out all the data for an entire animal at a time, or several of the cows, fit the model to a subset of the data and predict them, and see whether the overfitted model with unstructured covariance or the two other models with more appropriate covariance parameters do better. I'm not going to run that code here because there's not really a way to save the pre-fitted object as easily, and it actually takes, I think, several hours to run, because it has to, actually, I did 5-fold cross-validation, so it has to refit each of the 3 models 5 times, and each one takes a couple of minutes. So it takes a long time to run. But just I'll show you the results in the slides. We're grouping by animal, dividing up the cows into 5 groups, fitting the model to 4 of the groups, and predicting the 5th group's data each time, and seeing which model does the best. So a model that fits the data well but doesn't have too many parameters and isn't overfit is going to be the best at predicting unseen data that it wasn't used to fit that model. What do we see when we compare these 3 models using this very robust criteria of cross-validation? Yes, so in the slides, I've shown the output that you would get if you sat here for an hour and ran that code. The AR1 model is the best fit to the data. The unstructured and compound symmetry, this is like the difference. This is the difference between the quality of the model fit and the standard error of that difference. And sort of like a difference in AIC, if you're familiar with that. Compound symmetry turns out to be a very bad fit compared to AR. And the unstructured, again, it is a closer fit to the actual data, but it has so many extra parameters that it was overfit and doesn't do well in cross-validation. So the AR1 strikes the best balance between simplicity and how well it fits the data. Again, because these really are repeated measures from the same cow in multiple days, you would assume that they are more closely related. The closer, if you knew the cow's weight at day 50, you'd probably be able to predict its weight at day 45 back in time, but not as well its weight at day 20 or something like that. So that's for these type of data, AR1 is often a good choice, and our cross-validation shows that. Any questions about that? So the last things we're going to do with our cow model is now, that was just sort of a proof of concept to show you how the residual covariance works. we'll do what we would normally do if we had a data set like this and actually make inference about the treatment effects, since that's what we're interested in. Does iron help or hinder cows to gain weight, depending on whether they have tuberculosis or not? Yes, and oh yes, I should have mentioned this. So I think the reason the AR model does best is because it's a statistical model that best captures the actual biological process that's going on. Like weight gain of an animal over time is an autoregressive process. Its weight at one day depends directly on the weight at the day before. But if you have a process that's kind of more random fluctuations in time, like annual differences in yield, maybe compound symmetry will be better. Like the relationship between within a location, you know, the relationship between yield in 2 years, 1 year apart isn't that much more closely correlated than 2 years apart or 3 years apart, because it's more random fluctuations. It doesn't depend on the one previous. So then you might use compound symmetry. It might be a better fit. But here AR is a good fit because it's actually capturing the biological process that's occurring.
Quinn,
Yes.
sorry. Just before we move on the autoregressive code, can you remind, I know you said it, but I've already forgotten, I need to annotate it, what the P=1 addition in that line is doing?
That is the default. That's actually the default, but I just put it there to be explicit. It's a first order autoregressive. Like when we were doing our SAS model, we put type equals AR parentheses 1. You could put AR parentheses 2 or 3 if you wanted to, and it means the covariance matrix has a different form, and there's like extra parameters for higher order distances between time points.
Okay.
Honestly, I rarely would go to that.
Yeah, could you level. I didn't even know you could. I've only ever done AR1. Could you put an H in there for like heterogeneous autoregressive, or is that a different one as well?
Yes. There's I'm not sure how many different covariance structures BRMS supports. It probably does. I'll have to look up the syntax. If you really wanted,
in SAS I've learned like SAS, 4 or 5.
Yeah, that's one thing about SAS. It does support a wider variety of built-in covariance structures than a lot of R packages. BRMS actually has most of the ones you would really need in most cases, but it might have that. I have to look. If you really wanted a customized one, the solution here would be to write your own Stan code with your own customized covariance matrix, but that's sort of a one you'd probably have to call me up to help with that one.
Yeah, I think so.
You know, again, it's like if it helps fit the data better, good. Otherwise, it might be needlessly overcomplicating things. But yeah, I think it can, but I'd have to. I don't know off the top of my head.
Well, and most of the time I wind up using in SAS AR1 anyway, just because like what you just said in the slide, most of the data I'm working on is like yield data, but taken over subsequent weeks or months, and because it's dependent on the previous time, it usually winds up fitting best anyway.
Totally. Yes, we can get into like some really esoteric covariance matrices. The ones I showed are just a kind of some of the simpler ones, but I mean, any combination of like ways you could make that matrix, you know, you could do it in almost any way you want. It just depends on how well it fits the data.
Yeah, thank you.
For sure. One thing I did also want to point out, I forgot to mention this as well, is that if you would try to fit an unstructured covariance matrix to this using like a frequentist procedure, like NLME and R or proc glimix and SAS, it would not even close to give you an answer. It would completely not converge. The Bayesian approach actually lets us even get an answer for the unstructured covariance model because of the regularization. It shrinks those parameters towards 0, so that even though the model is complex and has hundreds of parameters, it's not too overfit. So actually, even for the unstructured covariance, the Bayesian approach does decently well. Like I tried to fit this model these same models in frequentist and could not get the unstructured one to converge at all. And yeah, that's because Bayesian can handle models with a lot of parameters better because it's putting in that regularization and not overfitting even when you have a lot of parameters, which is kind of cool. Yeah, so just before we take a break to finish off this example, just in the interest of making this similar to a real life analysis you would do, let's actually look at the treatment differences. We can make inference after we've now accounted for our repeated measures by cow as sort of a way to deal with that unwanted residual correlation in our data. We've accounted for it. Now we can, averaging over that, look at the predictions for each treatment. Let's say we wanted to know the rate of weight gain over time. How did it differ between treatments? We can use this function called EM trends, estimated marginal trends, to get a slope for each of the 4 treatment combinations. Like the 4 thick lines we showed on our graph before, do they have different slopes averaged over the random effects? And we can do, we're going to do reverse pairwise contrasts, contrasting the iron supplement versus no iron supplement within each level of infection. So pipe infect, meaning in the infected animals, is there a difference in the slopes for iron versus no iron? In the non-infected animals, is there a difference in the slopes for iron versus no iron? And the slopes we're going to get are still in those standard deviation units. We're going to go back later and convert them into our kilograms per day units that would be more interpretable. And so we use this, and we say var equals day after start. This is the name of the time variable that we want the slope for. So we want the contrasts between each iron level within each infection group, and we're going to use day after start. And you can see we're using the AR1 model to make these predictions. Since we've gone through this lengthy process to figure out it's the best model, now it's the one we're going to use to make predictions and actually make inference. EM trends is the name of the function, EM trends. We want reverse pairwise so that we're going to subtract our control mean from the treatment mean. Iron is the name of the thing we're comparing, of the treatment we're comparing. And infect, we're going to compare within each infection separately. And then var day after start. And I hit Control Enter. AMT underscore Diggle is what I called this. Diggle is the name of the author of the data set. If you look at this, EMT underscore Diggle, estimated marginal trends, you can see here we have the, if you go up to the top, this is the trend of how much weight gained per day in standard deviation units for cows that were not infected, that did not get iron supplement, and its credible interval. And the same for all the other combinations. Then the contrasts are the difference in weight gain rate between iron and no iron for non-infected cows and for infected cows. We can see the estimate. It's hard to know because we're in standard deviation units, but it's very close to 0, and the credible interval is quite symmetrical, about 0. So that's not very much evidence that there is a effect of iron supplementation on weight gain rate in either non-infected or infected cows. Again, do we want to, let's get some Bayesian style p-values to if people want that as to test the null hypothesis that there is no difference here, that this number is 0. We can use describe underscore, I'll use tab to complete, describe underscore posterior. And there's an object called EM trends inside of this EMT Diggle object. We'll use that, and then we don't want the credible interval since we already have that and we're going to just do the test. We're going to use the probability of direction and the p-map, the two different types of Bayesian p-value we discussed before. Control Enter to run that. Let me see. Oh, I'm sorry. We don't want the EM trends. We want the contrast is what we want the p-value for, because we wanted to look at whether this difference is different from 0 or not, the difference in slopes. This is actually testing whether the slopes are different from 0 or not. Obviously, they're all gaining weight over time, so we have very strong evidence that the slopes themselves are positive, but that's not necessarily interesting because that's not really biologically interesting, right? We want to know, is the iron supplementation making a difference in the slopes? So it's contrast that we want. But here you can see that we don't have very strong evidence for the for a difference. The p-values are, oh yeah, I should have added this. If you add comma as underscore p equals true, it will give the probability of direction as a two-sided p-value as well. So both of them will be more familiar to people that are looking at p-values that are lower, meaning stronger evidence against the null. Yes, so we don't have strong evidence based on this that iron supplementation is affecting the rate of weight gain in these cows. Again, it's a small sample size, so that could be, it's a low-powered study anyways. Before we take a quick break, let's just go through. Again, I'll mostly just save this for you to peruse at your leisure, but how would we write this all up? What would we put in a manuscript? And yeah, basically, this one is a little bit more involved because I would include in the methods section all the stuff about how you chose the appropriate model. Like if you tested different covariance structures and looked at the model fit, that's not really the objective of your study. It's more methodological. So I would put that right in the methods and explain the difference in the model fit statistics and everything. That's what I'd put it in the methods. And then in the results, you can just cut straight to the chase and say what is the evidence for or against the treatments having an effect on weight gain. And you can put in the Bayesian p-value if you want. And the credible interval as well. And in this figure, again, it's a lot to explain how it was created, but basically we want to generate the model predictions over time. So we generate a model prediction for every combination of treatment and a grid of evenly spaced days to plot it. And then I rescaled, I sort of undid the transformation where I turn weight from its standard deviation units back to kilograms per day and the days after start to the actual day that was in the original data. And so let me just copy this code and show you what that looks like. And we have to say include autocore equals false to give us the average prediction, not the individual predictions that account for the autocorrelation within each cow. Although they're accounted for in our sort of inference, it'd be too complicated to plot them on the plot. So we just want to show the averages averaging over that autocorrelation. So it takes a little time to compute all of that. So you can see, and I just kept the original the original trends for each cow behind the data. You may not want that because it's too busy, but I do think it's nice sometimes to show that the model is a reasonably good fit for the data. So you can see here are the trends. The slopes diverge a little, but apparently not enough to be significant in terms of iron supplementation. Now, this isn't the perfect model. You can see there is a little bit of a relationship between the mean and variance here. When there's low weight, the variance is lower, then it goes up. So really, like, maybe we should have assumed a slightly non-normal error distribution or transformed the data or something to account for that. But we did show the model predictive check plots looked like it was capturing the shape pretty well. So, you know, maybe that isn't too important in this case. It also looks like maybe the trend is a little bit nonlinear, like the weight is increasing faster and then leveling off. So maybe this just a plain linear slope is not ideal. Again, these are just things you might try to play with if you were really working with this data in real life. Although this is actually doing a pretty good job, I think, of capturing most of the pattern that's going on in the data. And I also have included some code to make a table of the slopes and convert them back to kilograms per day units. So I'll just show you that as well. I'll copy and paste the code, and then I'll explain what the table shows. Yeah, so you can see that I've included the 4 treatment combinations. This is the slope back in units of kilograms per day. So this is actually an understandable unit. And I've given different credible intervals for them, 66 and 95, which I often like to use because 66 is roughly 1 standard deviation and 95 is roughly 2 standard deviations. And, you know, just looking at it, eyeballing it, it's like, well, they're all around between 0.3 to 0.4. You know, yeah, the infected animals do have a lower slope. I didn't do that hypothesis test. It would probably come out as a more significant difference. Infected versus non-infected does obviously have a pretty big effect on the weight gain, but the study was kind of set up to look at the difference of iron supplementation. So that was what I focused on. Yeah, so that concludes this example. What questions does anyone have before we take a quick break?
Hey, Clinton, this is probably a stupid question, but
No.
what's the
Not at all.
significance with the posterior distribution contrast? What's the significance of the medians being negative versus being positive?
Let's see. Yes. So
Because I noticed before you corrected it, the medians were all positive, and then they turned
yes, so
negative
this
for
is the difference in the slopes. So the slope of the line for iron is a little bit smaller than the slope for no iron. So that's why the estimate is negative. It's just like
I see.
the difference in the slope.
Okay.
So that could either be negative or
see.
positive, depending.
I see. Okay.
Yep.
Gotcha.
So yes,
all right,
but even though the median is technically negative, it's very close to 0, and the credible interval straddles
that's
0
why the other
quite a bit.
medians were
So
positive, just because the slopes were continuously increasing with the weight. Right, basically, the slopes are positive because the weight was increasing.
That's right. Yes, the slopes are
Okay.
all positive because you know, they're clearly highly positive. So that's why I did
Okay.
I wrote the wrong thing in there by mistake. This was not what I intended to test, but it does show us that if you did want to test that the slope is greater than 0, you could. But
Gotcha.
obviously
Okay.
they're all gaining at least some weight, so the slopes are positive.
Okay. All right. Thank you.
Sure. And I see Grant has left a comment that there's some more documentation about different covariance structures. So if you're getting into like the heterogeneous stuff, then, you know, and also the AR1 does assume that the time points are evenly spaced, which actually in this data set, they're not quite. So really there's other covariance structures that deal with slight differences in the length of the time gap between measurements, but I didn't get into that because I thought it was excessively complex for this, just to introduce you to it. Yes, Marina, that's right. In this example, it looks like anyway that iron supplementation was, even if anything, reducing the weight gain. Yeah, but I wouldn't say iron supplements reduce weight gain, right? Because we didn't have evidence for a difference. There's sort of evidence for technically the median is a bit lower of the slope for iron, but there's just not strong enough evidence to say, I wouldn't say reduces non-significantly. I would just say there's no evidence to say that they help or harm. But yes, certainly there's no evidence they're helping, right? So there's that. Great. Let's take a 5-minute break and come back at 4:17 Eastern. Yeah, see you in 5 minutes. I will be right back. Here we go. I'm getting some good tips on ergonomics for the workplace, and people are having a good discussion about how to interpret results of statistical models. So that's great. Oh yeah, and I again referring to Grant's comment. the BRMS documentation refers to phylogenetics. So that's another case. I didn't really mention this, but a case where you would use covariance matrices in a mixed model would be in like a phylogenetic or quantitative genetic context, where like you might have a covariance matrix based on the genetic similarity between individuals in your data set. So like two individuals that are closer in, their sequence similarity is closer, their genetic distance is lower, will have more correlated residuals. So that's another case where you would use this, obviously for like breeding and stuff like that. It does come up a lot. So yeah, that's another case where you would need this approach. And as Grant was saying, you could also, instead of using the model to estimate the covariance matrix, you could actually get the covariance matrix externally, like from genetic relatedness, and put it into the model without using the model to estimate those parameters. It can be done with BRMS as well. If anyone is interested in that, we can discuss that later. Okay, I will go ahead and we'll get through whatever we have time for of the last example in lesson 3. Maybe we will get all caught up by the end of today. If not, it's cool too. I said before that there's 2 ways to deal with modeling relationship in space and time. One is to put it in the residuals. Another is to actually put it in the model itself. So let's look at that second way using a spatial, a situation where we have variation in space instead of time. And we're going to use a spatial additive model. So I'll give you a bit of background before we go and talk about the example. So yeah, again, we're not just throwing it in the residuals, we're actually modeling space or time directly as one of the effects in our model. We can actually make inference based on it then if we wanted to. The simplest way to do that would just be to add a linear predictor term in your model for the effect of space or time, or both. But often that's not enough, right? Like you would add latitude as a fixed effect and longitude as a fixed effect, for example. But that's only going to allow like a linear trend in each, north-south versus east-west. Spatial effects don't often work that way. They're often not linear. So what do we do in that case? Well, what you do is go and fit a generalized additive mixed model. We're going to model that response variable as being a function of some combination of unknown smooth functions of the covariance. So this is a spline or a smoother. I'll show you an example of that in a second. It will make more sense. And there's many different types of splines you could use. We're not going to get into that can of worms very much right now. But what you need to know is it's kind of trying to fit a squiggly line to the data, but not too squiggly. There's a built-in, it sort of has its own, in addition to the priors, it also has its own built-in penalization process to fit the data well without overfitting it. And I'll show an example of how that might work. There's a parameter called K, which is the basis dimension of the spline. That's basically how many squiggles can the squiggly line have. It's like the maximum it could have, and then there's a regularization that goes on. So here's an example of a simple generalized additive model fit to a time series data that's not linear. This is like data from a crash test dummy accelerometer of a dummy that was in like a car or motorcycle crash, where it suddenly decelerates when the crash happens, and then it whips back and accelerates again before it finally stops. And right, so it's not linear, right? It's sort of flat, goes down, goes up, goes down, and is flat. So basically this GAM is trying to fit a combination of polynomials to the data. And this K number governs how many different polynomials are being added together to produce the final fit. If it's 5, this is as close as we can get. You can see it's not really a very good fit to the data. If we set k to 15, here's what we get. We get quite a good fit to the data. You see it's capturing the main squiggles without trying to go through every data point. What if we set k to 75? Here's it. You can see it actually looks almost exactly like the k equals 15 because of the internal regularization that is being used by the GAM process. I'm not going to get into the details of how it works, but it's basically keeping it from trying to go through every data point. It's compromising between fitting the data and not fitting it too closely. So really, you can set this k value as high as you can. As the higher you set it, the more computational time it will take to fit the model, but you can set it really, it doesn't really matter. You don't want to set it too low because you'll miss important information in the data. But if you set it too high, the only thing that will happen is it'll take longer for the model to fit. It is actually controlling already for overfitting. Does that make sense? Please give me a thumbs up if you follow my train of thought. Or if you have a question, please ask. All right, thanks a lot, folks. I've put some resources for further reading in the lesson text if you want to learn more. Again, I'm just giving you very big picture stuff right now. So this, if you're a Scotch drinker, this is a good example data set for you because it is a field trial of barley varieties used to produce whiskey in Scotland. And there's, it's, a classic agronomic study where we've got several dozen genotypes, and we wanted to look at different yield, if it differs between the genotypes, and also it was a study testing the effect of 2 different fungicides, whether that affected yield. So we have a full factorial with 2 fungicides by 70 genotypes. It's a randomized complete block design with split plots. That means we've replicated every treatment combination 4 times in 4 replicate blocks, but we have a main plot and a subplot effect where each block is split up into 2 halves. One half got fungicide and the other half did not get fungicide, and each of those halves is split into a little tiny subplot for each genotype. This type of split plot randomization is often good for agronomic studies because if you're spraying fungicide over a large area, you can't randomize it at the level of a tiny subplot because you can't spray on a tiny area without the spray, getting away from the plot you're trying to spray. So but you could sow different genotypes at a very fine resolution. So we do the randomization of fungicide spraying at a larger physical size than we do of the genotype. That's a split plot. And we're looking at yield as our response. And the way this experiment was set up, we have 4 blocks with 2 main plots, one with one type of fungicide and one with the other. So there's 8 of these plots, each of which has little areas for the different genotypes. They're all in a long line, sort of an 8 by 1, and each of the blocks, each of the main plots has rows are the going across, and they call them beds going up and down. So that's the name of the columns in the data set, bed and row. And I've just made 3 different plots to show you what the different blocks are. I've colored it by block. Then I've colored it by fungicide treatment. And I've also colored it by the subplots, the genotypes. And I've put the aspect ratio 10 by 56 because that's the actual shape of the study area. So the dimensions of the figure will look like the actual dimensions of the physical field in real life. So we're at line 94 now. We'll run this data function to import the data into our environment from the example data in the package. If we just type head parentheses Durbin et al. Is the publication this comes from, Durbin.splitplot, and hit enter, you can see these are they're pretty simple. Yield is a continuous variable. We have blocks, we have genotype IDs, fungicide 1 and fungicide 2, and then our X and Y coordinates of each row and bed of each one. So we're going to plot X is bed, Y is row, and we're going to color it in by block. Hit Control Enter, and you can see there's 4 blocks, all in a row. If we color it by the column is called fung for fungicide, that'll show us where the fungicide treatments are within those blocks. Control Enter. You can see they are randomized whether the left or right side of each block got the fungicide treatment 1 or 2. And let's fill by genotype. You can see that there's just a bunch of genotypes in little square subplots within each of the fungicide areas. Now let's look at what does the response variable of yield look like when we plot it on a heat map. And I put this in a single color palette of different shades of orange for the yield, low versus high. It's nice to do in one color, so it's colorblind accessible, or it also prints in black and white as well. So when we look at the yield, you can kind of see, if you look at the figure, you can kind of see those lines separating the fungicide one from the other. So it seems like one fungicide tends to have higher yield. But you can also see some spatial patterns in the data. For example, this area in kind of the top has a lot of low numbers near each other. So maybe that is an area which has poor soil, or, you know, the drainage is different there. So the yield is different spatially. Even after accounting for like the treatments and genotypes, they're low yield over there. And like in other areas, here is a cluster of high yielding plots, which could not be explained alone by treatment and block and genotype. So there's spatial patterning in the data. What if we, this is just for demonstration purposes, you wouldn't do this in real life, but what would it look like if within each block and main plot, we randomly shuffled around the spatial locations of the genotypes? Would it look more like if there was no spatial pattern and we randomly shuffled this within each main plot, it wouldn't look any different than the actual distribution of the plots. So just as to prove to ourselves that there really is a spatial pattern, let's do that and randomly shuffle the locations so that we can see what it would look like if the yields were shuffled randomly within each of these 8 areas. And one way to do that, I've set a random seed so that if you do this random shuffling, it will get the same result as me. You don't have to set the random seed, but you can. Group_by is how we want to do the sampling within each of the 8 combinations. That's the combination of block and fungicide within each of the 8 main plots. And sample means randomly sample the values there. So I'll highlight this and I'll run that code. Now I've assigned that to a new object called Durbin splitplot underscore shuffle, because we're not going to be using this to fit the model to. It's just to make this plot real quick. And we're going to do fill equals yield again. Same plotting code, but using the shuffled yield values. What does it look like? Let me see. Yes, so yeah, wait a minute, that doesn't look different. Where's the just make sure that I did that correctly. Oh, yield underscore shuffled. I didn't change that name. So that was the unshuffled yields. Thank you, Caitlin. There we go. Yes. So once you do that, you can see that we still see the difference in the fungicide treatments is visible, but we don't see as much of a spatial pattern. Certainly, we don't see those as much of a cluster of low versus high yield within the blocks. So it's kind of a visual evidence to me. A little, like, go back to this one. You can see there's more spatial pattern there than this one. Looks more random. So yeah, there's something spatial here that isn't going to completely be modeled if we just modeled the blocks and treatments and genotypes. Okay, does that make sense to everyone? We still have a bit of time here, so we should be able to get through at least most of this. First, again, as I usually do, I've sort of showed you that it's important to model the spatial component, but let's prove that by making a model without the spatial component accounted for and see where it breaks down. So our response variable, I'm going to do a scale transformation, a standardization of the yield variable within the model. I'm going to convert yield to standard deviation units just inside of the model. Because that will help us decide what prior to put on the data without having to worry about what units yield is in. We're just going to look at standard deviation units. And our fixed effects, right? We have genotype, GEN. We have plus fungicide, FUNG, plus genotype : fungicide for the interaction between genotype and fungicide. Of course, the shorthand for this would be genotype * fungicide. I've just typed it all out here, although you could use the shorthand. And since this is a split plot study, experimental design, we have a random effect for block. So each of the 4 blocks gets its own random intercept. And then because we have many subplots within each main plot, and those are the eight main plots, which are identified by each combination of fungicide and block, we're going to give a random effect at that main plot level as well. Fungicide : block is our other random intercept. And I've just decided to put a prior with a mean of 0, standard deviation of 3. 3 standard deviation units is a pretty weakly informative prior because it puts the most probability on effects near 0, but has a decent probability of pretty large effects. 3 standard deviation effect of yield would be quite large. And B, because we want the prior on the fixed effects. And I've increased the number of iterations on each of our 4 chains. We're going to do 2,000 warm up and 4,000 total. So we're going to have 2,000 after warm up for each chain. And in my testing, I found that was enough to get convergence. In real life, you would have to play with it and see how many iterations are needed to get convergence. But I'm just take my word for it that is adequate. Control Enter to run that. Okay. Now, we want to show that my goal here is to show you that this model is not appropriate because there's still a spatial pattern in the residuals. Residuals are just supposed to be random noise that's left over after you've accounted for everything that you need to account for. If there's a pattern in the residuals, that means there's a variable that should have been in the model but isn't, which in this case is the space effect. So let's just, as a proof of concept, we're going to pull out the residuals from this model and plot them and see if there's a spatial pattern in them. So we're going to use the function residuals, which is a function that works on most statistical models in R and pulls out the residuals for every data point. the error that's left over, the difference between the model's predicted value for that data point and the actual observed value for the data point. And we're going to pull it out for the Durbin LMM, linear mixed model. We're pulling out the residuals. Control Enter. And then we are going to add those residuals as a column onto our original data set so that we can plot them the same type of map as we did for everything else. So I'm going to mutate the original data set, the Durbin split plot data set. I'm going to make a new column called resid no spatial, residuals from the model that ignores spatial variation. And we, you actually, this residuals object has multiple columns because it has the point estimate and the credible interval on the residuals. We only want the first column, the point estimate. So we want bracket comma 1. That's going to take the first column of that residuals object. Since we don't really need, we're just trying to plot this. We don't need to worry about the variation in the residuals across the different posterior iterations. We just want the one estimate to plot. Control Enter to run that. Now we can plot the y, the fill variable that we're plotting is this column, which I named resid underscore no spatial. Other than that, it's the same code. I just decided to make the color green this time. Hit Control Enter to run that. So when you look at this, you can still see some spatial pattern in the residuals, especially like here. There's that area of low yielding plots, which we still, it kind of spans multiple blocks. We still see that even after we've accounted for block and treatment and everything. This area has a negative residual, so the yield is the residuals are lower than we would expect there. And you see this area, for example, in the lower right looks like higher yield even after accounting for everything else. That's a pretty good way to do it, is plotting them spatially. There is a statistic you can use to measure how much spatial autocorrelation there is in the residuals of a model. In other words, what is the correlation between any two residuals that on this map and the distance apart they are? If those things are correlated, that means the closer together 2 points are, the closer their residuals are. That means there is a spatial pattern in the residuals. We do not want this correlation to be high because it means there's a lot of spatial pattern in the data that we're leaving out of our model. And that statistic is called Moran's I statistic. What we have to do is calculate the inverse distance between any 2 points in our, and we're going to count like each plot as being 1 unit apart, just the distance in units of plots, and then take the inverse of that, 1 over the distance. And so we have to basically make a matrix of the x and y coordinates of each plot, which is the bed is the x coordinate, right, on the map, and then rho is the y coordinate. Then we're basically combining those into a matrix using the function dist to get the distance between each pair, convert that into a matrix and take 1 over that. And then we have to set the diagonal of that matrix to 0 so that the distance between any plot and itself is 0. I'm going to run this code here. And then I'm going to look at the Moran's I statistic. The function is from the ape package. It's called moran.I, capital I. And the first argument is the thing that is the residuals, the thing we want to see if they have spatial correlation. Resid underscore no spatial is the name of the column with the residuals. And the second argument is the inverse of the distance matrix between each pair of those residuals. Durbin underscore inv underscore dist is what I called it. I'll hit Control Enter and run that. And we can see the correlation is 0.04. That's in an absolute sense, it's not super high. In practice, you could probably, the rule of thumb would be like if it's less than 0.1, it's you know, not that big, but and you could possibly get away with ignoring it. But the idea is we are trying to show you know, the importance of accounting for space. So we will go ahead and fit the spatial model and see if we can improve on this. And oh yeah, it gives you a p-value as well, which is so small it can't be distinguished from 0. So we have very strong evidence that even though the magnitude of the correlation is 0.4, we have very strong evidence that it is different from 0. Please give me a thumbs up if you're still alive and are still following this. Okay, yes, that's more than I thought. Great. Well, you know, I think that it is basically the rest of this example is fitting the spatial model that accounts for the spatial fits a smoother to the spatial term, and then we look at whether or not that accounts for the spatial pattern in the residuals and whether that has any influence on the conclusions we draw. But it is 4:45, and I think that we've done a lot for today. So I think we can save that exciting cliffhanger for next week. We can actually fit the GAM and see how we do. So what I'd like to do is if have people ask any questions they have or any discussion they want to raise for the next few minutes. So yeah, the floor is yours. Is there an easy way to calculate the inverse distance when plots are non-square? Yeah, I mean, I think you would probably have to estimate somehow the location of like the center of each plot in some unit of meters and then do it that way. Yeah, I mean, honestly, I'm not even sure if the plots in this data set are even really square, but I'm just assuming they are for demonstration purposes. Right, if the plots were like 10 by 1 or something, then you're assuming they're square is going to really throw off your inverse distance calculation. So yeah, I would say, you know, you'd probably want to like you could roughly estimate it if you knew the dimensions of the plot. You could just multiply the coordinates in plot units by the length and width of the plots in meters, and that would give you, you know, because there might be like alleys too, which would like change the distance a tiny bit. It's probably not going to strongly influence the results, but if you were concerned about it, then certainly the best thing to do is to actually get the real coordinates of the center of each plot. Yeah. Good question, though. Anyone else?
Quentin, I have a question.
Sure.
So I guess this split plot example, this Durbin one we were just working on, right? When you're doing this, you need to calculate these values down to the smallest like experimental unit, correct?
Yes.
Okay.
Would. Yep. So yeah, you know, we physically know where each subplot is in space, and they have they have their own individual yield measurements. So that's the ones we would want to. The residuals are at the level of subplot. So that's what we, you know, the number of, there's like 560 observations. Those are the individual subplots, and we get a residual for each one. So that's what we have to also get the pairwise distance for every pair of those 560 subplots. Yep.
Okay. I'm just trying to apply this to my own studies where I'm doing
Yep.
something
Yep.
similar.
Yes, and I'm happy to consult with you on that whenever. Great. Well, I will stop the recording here. Your folks are free to go whenever. But I will stick around and answer any more questions. And just to note that I have been putting you should be able to see the recording afterwards in Teams if you go into the meeting and click on recap. But I have been posting everything on the Axon page, which I'll just throw a link to that in the chat real quick. Yes, thank you for being here. I'll throw a link to that in the chat. Oops. So yes, that page has all the videos on it, and I should get the video for today up there sometime tomorrow. Yeah, so thanks again to everyone for being here.